Hello. Today we're going to be talking about mitochondria quantification in human embryos during embryo development and relationship between mitochondria load and Ploidy status. My name is Nabil Arrach. Disclosure, I am the CEO of Embryogenesis, a company that does Ph.D. and PGS. Okay, well, this is a classical table that shows a correlation between implantation rate and chromosome abnormality. The implantation rate goes from 31% under 35 years old to 6% in the 41-42, while the chromosome abnormality goes from 54 to 82. So as we age, the implantation rate decreases and chromosomal abnormality increases. And so that's why we do pre-implantation genetic screening to increase the chance of getting a normal embryo that will implant. So on average, in the United States, the success rate for implantation is approximately 70%. And so that is on the high end. So the question is, what happens to those 30%? Why aren't we able to get a much higher success rate in implantation? So we've been, in the course of IVF work, we've been looking at morphology of the embryos, going from morphology to chromosome copy screening to time-lapse. We have looked at metabolism of the embryo, and now we are looking at mitochondria. And the question is, does the mitochondria add any value to the embryo screening to increase the chance of implantation? And so the three main points I will try to make in this presentation is the correlation between mitochondria load and Ploidy, mitochondria measurements and in conjunction with time-lapse, and whether mitochondria is something we should look at to improve our implantation rates. A little background on mitochondria. So mitochondria contain, human oocyte contain approximately 100 to 200,000 units of mitochondria. The mitochondria is not made in those 5 or 6 days of embryo development. It's essentially distributed while the embryo is dividing. And so the embryo gets less and less mitochondria per cell as it goes from day 1 to day 6. Mitochondria has a mutation, a higher rate of mutation, 10 to 20-fold higher than nuclear DNA, and mitochondria is a main source of ATP synthesis, which is required for cell to function. So in our study, we have looked at 325 embryos, day 3, day 4, and day 5. And so what we were looking at these embryos is the mitochondrial load, and we were also looking at the Ploidy status, and down the road, looking at implantation rates. And so in this figure, you will see that when we look at, this is day 3, day 4, and day 5, so age of the embryo. Those are the ratio mitochondria versus nuclear DNA. In blue is euploid and aneuploid. We're looking at all age groups. And what we see is 2 things. First is the mitochondria decrease over time from day 3 to day 5. And the second observation is that euploid tends to have less mitochondria than aneuploid. If we break this data into 2 age groups, younger age group, 32 to 37, we see the same trend, decrease of mitochondrial load, and also we have less in euploid and aneuploid, and we see the same thing if we look at older patients. So remember, those embryos are the day 3, day 4, and day 5 are not taken from the same embryo. Those are groups of embryos that are taken either at day 3 or day 4 and day 5. They're not connected. And so if that data, because we're looking at the median value of all these 325 embryos, we're hoping that at some point we're going to see some trend that reflects most of the embryos in that group. So if data was continuous, you would see that this is the mitochondrial load. This is another way of looking at the data, mitochondrial load ratio, and this is day 3, day 4, day 5. In blue is the contents of mitochondria in euploid, and this is aneuploid. So what we see is that the mitochondria load decrease at a much higher rate in euploid compared to aneuploid. And the question is why? Why euploid embryos will get rid of the mitochondria at a much faster rate than aneuploid? So if we put time-lapse into this equation, what we would see is if embryos are dividing at a lower rate, so abnormal embryos will retain more mitochondria per cell on average than euploid, simply because they don't divide as fast, so they will retain more mitochondria. So to look at that question, we tried to look at the same embryo on day 3, on day 6, both using time-lapse and also mitochondria quantification. So we looked at 39 embryos cryo-preserved from zygotes to day 6, and we did time-lapse, and we looked at chromosome copy as well as mitochondria. So the results. So we see that day 3 embryos still have very little mitochondria compared to day 6, which confirm what we have seen in that large study. Also, embryos that have arrested embryos tend to accumulate more mitochondria than non-arrested embryos. These correlation that we've seen confirms that euploids tend to have less mitochondria than aneuploid. And this was tested using a statistical model. And so this is a subset of the data. So you look at, when you look at these embryos 1 to 14, and this is just a score, MitoSure, that we use, but really it's just a simplified form of looking at instead of looking at 0.0001, we look at higher scores, so it's easier to compare. And so this is embryo, is at the embryo level. So we took the entire embryo and we looked at mitochondrial content. So green are the embryos that have arrested prior to compaction. This is time-lapse data. And the red ones, most of them are continues to grow till expanded blastocyst. And what we see is the one that arrests, when you look at the mitochondria in the embryos, they are much higher compared with the one that don't arrest. We looked at the pregnancy rate coming from the first large group that we have analyzed, the 325 embryos. And so, we looked at the embryos that were transferred at day 5. We see that most pregnancy happen at a very low mitochondrial load. And the observation we made in that course of looking at the data was two things. We have seen that that embryos with very high mitochondrial level never implants, and the one that fails to implant are not all mitochondrial deficient. Some of them are maybe high in mitochondria, but some others are not necessarily having any issue with the mitochondria. And that tells us that there are more factors that plays into the implantation rates beside mitochondrial DNA. And so the question I would like to raise is, when we see an embryo that fails to implant and it has high load of mitochondria? Is it because of the mitochondria is causing a problem to the embryo, stress, oxidative stress, or any other type of issue that prevent that embryo from implantation? Or mitochondria is simply a marker, a reflection of how fast the embryo is divided? In our future directions, we're going to focus on a large study doing more time-lapse, this time in a large study, and we're going to try to look at non-invasive ways of testing embryos, blastocell, both for mitochondria and Ploidy status. And I think with that, I would like to thank you for attending this presentation. This work has been performed in collaboration with USC Fertility, Center of Advanced Genetics, and RCMC. Thank you. If you have any question, please ask.
Thank you for the excellent presentation. And now the word is free for the audience, please.
Thank you for that presentation. I wasn't entirely clear. For a lot of the presentation, you're referring to mitochondria, but are you actually talking about mitochondrial DNA or mitochondria? Are you talking about the organelle itself? When you say lots of mitochondria, do you mean lots of the organelle or lots of the DNA?
DNA, because the only thing we can measure is the DNA sequences, and we're assuming that DNA sequences are reflecting the amount of mitochondria units.
Okay, and how are you measuring that?
We're doing a ratio of mitochondria sequences obtained in next-generation sequencing compared to nuclear DNA sequences.
Okay, so your data is generally concordant with the other data from Friguli and other people over the last couple of years. Okay, and are you applying this clinically yet?
We are using, we have had large samples that we have monitored for pregnancy outcome and all that, and we are providing the information to clinicians to be able to at least have an independent way of looking at the embryo. But I believe we still have to do more work especially to gather more pregnancy data to be able to tighten up the correlation between the mitochondria load and pregnancy and implantation rate.
Yeah, I mean, certainly I think you'd need a lot more than 300 embryos before giving clinical advice. Yes. Okay, thanks.
Yeah, thank you.
One more.
Schmutzer from Kiel, Germany. Would you agree that the message from the CoGen meeting in Paris last year was that the truth in PGDIS in PGS is not black and white, but we should make a ranking of the embryos? And I think your results go in the same direction, so that we could rank all embryos and then decide which one is the best.
Exactly.
There's not the truth, and this might be advance and also persuade the enemies of PGS that the truth is not black and white.
Right. Exactly, and I think as we see more information coming from time-lapse and the selection process using a number of parameters in time-lapse, I think that's in conjunction with the Ploidy and mitochondria and perhaps additional markers will help us make the selection process a little better and choosing the best embryo possible for a good outcome. I agree.
Mitshiwi, USA, Nabil. Thank you. When you said that the euploid blastocyst had lower mitochondrial content, were you comparing it to other normal aneuploid blastocysts or to all aneuploid embryos, including the developmentally arrested ones?
We looked at the 325 every time when we took the median value for approximately 100 embryos. And so when you look at the median values, you see clearly that aneuploid have more mitochondrial load than euploid. When we looked at single embryos, we see the same thing. Most of the time, there is a factor of twice more, sometimes more, mitochondria in euploid.
But you're comparing only blastocyst to blastocyst.
Blastocyst, correct. Because at day three we have a mix mixed data. We have data that shows aneuploid tends to have less, and other data shows the same. So I don't think it was conclusive. I think day 3, from my personal experience, I would say there is no major difference between the two. The difference happens at day 5, and I think because because the embryos are not dividing at the same rate, so the aneuploid tends to retain more mitochondria over time.
Thank you for your presentation.
Thank you.
Thank you.